Hi, welcome to Neuroverse, a podcast hosted by Carolina
and Clara, where we discuss all matters from neuroscience to philosophy and beyond. This episode is part of our podcast series, Tools for Studying Brain and Behavior, which is kindly sponsored by Scientifica, one of the world's leading producers of electrophysiology and imaging equipment. Be sure to check out the other episodes in this podcast series to find out more about methods for studying the brain, and also check out Scientifica's website for information about products, resources, events, and support that they provide for scientists. Before we begin the episode, I also wanted to mention that we did a previous episode, number 45, Measuring the Brain in Space and Time, where we discussed the methods used to measure brain activity and the important trade-off between spatial and temporal information. So specifically, we were talking about how neuroimaging methods prioritize spatial information, or they have higher spatial resolution, whereas electrophysiology techniques have greater temporal information, which means that electrophysiology is essentially the ability to measure electrical signals from biological substances. And this is often done in the brain, of course, where there are bountiful electrical signals. And so any form of recording neural activity in electrical form, so in terms of voltage or current, is considered electrophysiology. We hope you enjoy the episode. Today's episode is on patch clamp. Patch clamp is a technique, which is a type of electrophysiology and one of the smallest scale electrophysiology tools. And in today's episode, we're going to be explaining to you what patch clamp is, how it works, what it can inform us about the brain or neurons, and the different types of patch clamping techniques that are done. So this is a technique that's pretty close to the hearts of both of us, as we've both done it in the lab. So naturally, we know the challenges of the technique, which there is a nice long list of challenges, but it's also an amazing technique that can give us lots of cool information. So that's what I want to share with you today.
Exciting.
Yeah. So to begin, the patch clamp technique involves a glass micropipette, which is tiny. It's smaller than a single cell body in width, and it's filled with an electrolyte solution and then a thin wire or electrode that's bathed in that solution. So the technique involves forming a tight seal between the glass pipette and a cell membrane of a single cell, which can be neurons, but it can also be any cell type that exhibits electrical activity, which actually most cells of the body do. And once you form this tight seal with a very high resistance, which is at least 1 gigaohm, so it's known as a gigaohm seal, you can then clamp or control the voltage across the membrane and then measure the current passing through it, called voltage clamp, or you can clamp the current and measure the changes in voltage across the membrane, which is known as current clamp. And hence, you are clamping voltage or current on this patch of membrane, and it's called patch clamp. So it's based on all of the biophysics of neurons, which Hodgkin and Huxley were the pioneers of that biophysics, and they first did this technique in squid giant axons. And without diving into the biophysics, the major equation that underlies all of how patch clamp works and the information we get from it is Ohm's law, which is voltage equals current times resistance, or V equals IR. And it basically means that the voltage that is recorded from a cell is equal to the currents transmitted through the cell and the resistances that they face as they travel through the cell, like the membrane.
Yeah, like the membrane, and also like the width of the pipette. So like the larger the gap you have in your glass pipette, the lower the resistance. But then it's more challenging also to patch the cells with a larger seal.
Yeah, that's a good point. So there's the physiological resistances within the cell, which is the membrane, which you can't overcome, but that's useful information to find out through the technique. But there's also the resistances of the equipment, like the pipette mainly. And so this is a really key point for exploring what are the options we can have for the different types of patch clamp you can do and what it can tell us. So the main, most commonly used one is called whole cell patch clamp. And this involves rupturing the patch of membrane that you formed the seal on with a mild suction. So it's pretty cool because, well, you can use a syringe, but you literally do a little suction like that to get into whole cell mode.
Yeah, and there's a tube that connects to the pipette. So that allows you to then be able to apply that suction onto the pipette, which then, yeah, like Clara said, we literally just apply suction, and you start off by doing it quite slow as you're approaching a cell. And then once you form a seal, you do a very quick to burst the cell open, kind of to break into the cell. And it's very intuitive. It's like an art. You really need to have a bit of a feel for the cell, and it's kind of cool because you can definitely tell a little bit the cell quality, like how healthy the cell is, if it's a small cell, a large cell, if in your pipette you have the width of the tip correct, if it's the correct width for the cell type that you're trying to patch. You know, if it's too big, it'll just like consume the whole cell. If it's too small, you won't even break in. It's impossible to break in. Yeah.
Yeah, it sounds like kind of primitive, I guess, when you first describe it to people, because it sounds very unrefined, but actually, like you said, it's so intuitive. And this is going to link to what we're going to talk about later, which is robots. I know.
Yes, I also saw that.
But yeah, it's very like, it's an art, like you said, because there's so many variables, and it's only by kind of feeling that you can sense them all and understand whether you're doing it right, if you've chosen the right cell. Yeah. Mm-hmm. So anyways, but that's whole cell mode, which is one of the most easy to achieve ones. And it's often used in combination also with stimulating presynaptic partners of these single neurons that you're recording from. So that can be by direct electrical stimulation, by sticking an electrode into the presynaptic location, or through optogenetic stimulation, which is often done to map neural circuits these days. And it can also be used to measure the electrical properties of cells, like intrinsic excitability, or the spontaneous currents can be recorded to give us clues about the synaptic properties of the cells. For example, if you change the holding potential, you can record the excitatory versus inhibitory spontaneous currents, and this can give you a hint at the inhibitory versus excitatory connections and synapses onto that cell. But a major problem with whole cell recordings is that you're essentially creating a continuum between the inside of the cell and the pipette. And so, like I said, the pipette contains a solution which is full of ions at specified concentrations because you make the solution yourself. And so when you go into whole cell mode, you can cause cell dialysis, where ions or intracellular messengers like ATP decrease in concentration or change in concentration because they flow into the pipette and mix. And so if the solution in the pipette contains a different concentration of ions to the physiological composition in the cell, then the dynamics of the ion channels, which is dependent on ion concentration gradients, and which is what patch clamp is essentially measuring, they will be affected. And so there are many ways to overcome this, but ultimately you'll see that it's all a balance of pros and cons, as is always the case with techniques. So, for example, instead of breaking into the cell, you could do a cell-attached recording. And so this is done without rupturing the membrane. You're still forming the high resistance gigaohm seal with the membrane, but you just simply keep the pipette on top of the cell and measure currents from there. So you can study single channel currents or the sum of currents from all the channels in the patch of membrane. And this is often used to measure spontaneous cell firing activity also without changing the cytosolic content of the cell. But like Carolina mentioned, the point of whole cell mode is that the pipette resistance, which is the access resistance that you have with the cell, is very low. So the electrical access is optimal, which you don't have in cell attached mode. Another option, which doesn't risk cell dialysis, is perforated patch. This involves forming a seal onto a membrane, and you're not rupturing it. You're staying on the surface again, but the solution inside the pipette contains a pore-forming agent that permeabilizes the patch of membrane that it contacts. So it contains antibiotics that are pore-forming, and they integrate into the membrane. And this allows pores to form so that you're getting electrical access, you're decreasing the access resistance, not as much as whole cell mode, but more than cell attached mode, for example. But you're still able to use an antibiotic that is selective. So it only allows certain ions to cross the membrane, and you can choose a pore forming agent based on its selectivity. Depending on which ion you prioritize, that you need the physiological composition of that ion to remain the same, that's how you base your decision on which antibiotic you use.
I feel like, at least for the experiments that we've done in the past in our masters, it would have been a bit too medley. We would be interacting a bit too much with the different ions as opposed to just measuring the activity in the cell in general. So I guess if you want to target specific ions, if that's really relevant and specific to your question, that would be a way to do it.
Yeah, it also minimizes disrupting the concentrations of intracellular messengers, and these are really important for synaptic plasticity. So this technique is especially significant if you want to study plasticity, because often with whole cell mode, you're changing those secondary messenger concentrations.
Yeah, that's a really good point. Yeah, and regarding what you were saying earlier about how, there's a lot of problems about the internal solution that you have in your glass pipette where you insert the electrode to then patch clamp onto the cell and record the activity. The osmolarity and like all of the components are extremely sensitive. Like if it doesn't match the cell completely, then you won't get a good recording. You could burst the cell or the cell will die. The cell won't last very long. And I remember when I was making up the osmolarity, once we'd put it into the machine and it would like count down to see if I reached the correct osmolarity. We would just be like, fingers crossed, hoping they would work. And I came across this funny article about whether electrophysiologists are superstitious. Because we're so particular with all of our different techniques, and almost like we were talking about earlier, it's like an art, it's like an intuition, feeling how much suction to apply, and the osmolarity needs to be absolutely perfect. The seal size needs to be absolutely perfect. And I think it's funny, there does come an element of hope, at least a very strong element of hope.
Definitely, yeah. And talk about superstition. I think it's really interesting hearing about the different superstitions every patcher has. Like, for example, my personal one that I came to adopt was on the little pipette with the intracellular solution that I would use. I had to write a little smiley face or the day just didn't go well. And like, it genuinely worked. So it's all these little things. Yeah.
Amazing. I love that. I think it speaks to the challenges that come with being able to carry out a good patch.
I think it also speaks to the like how miraculous it is to be able to access a neuron and look at its activity in real time. Like, because that's possible, it's also possible that all these miracles can happen, that if your slice looks completely dead, you can still get a miracle and patch a neuron, like,
yeah, it's definitely magical. And also, you can also patch clamp axons and dendrites. And I recently found out that the way you do this is you actually cut the axon and this forms a bleb, and then you record from the bleb, which I think is crazy.
I think it's crazy because axons are so thin. I mean, so with whole cell mode, you're targeting the cell body. Same with perforated and cell attached as well. Dendrites are like hard enough, but axons are even thinner. And so I just don't really, I mean, it would be a dream to be able to try out all these different types. But yeah, I think axonal bleb recordings are particularly amazing.
Yeah, I had no idea that that's how you did it. Again, magical, how you can still have access to like the axonal properties through just cutting it and it forms a bleb.
Yeah. Some other challenging versions of patch clamp are also inside out or outside out patches. And this involves pulling a patch of membrane away from the cell and then measuring currents. And you can basically isolate single channels in this way. So for inside out, you retract the pipette, and this exposes the inner cytosolic surface of the membrane to the solution of the bath, and you can study in the intracellular side of channels. So this is ideal to study channels that are activated by intracellular ligands or to study intracellular signaling, because you can expose that intracellular side of the membrane to specific molecules and test the effects. And for outside out patches, the whole cell method is applied. So you rupture in the membrane and then you retract it. And this causes part of the membrane to detach from the cell and then reform into a small vesicular structure. And then with the outside of the membrane exposed to the bath, you can apply different drugs or molecules and then study channels and their extracellular properties and create a dose response curve, for example, for specific ion channels or receptors.
That's crazy.
Yeah. I also don't think that's very commonly done. Yeah, in the literature, really like a very large majority of studies use whole cell patch clamp. But then once you start looking at the different types of techniques that can be done, you start really seeing all of the flaws of whole cell. Like it wasn't until I really started doing perforated patch that I was like, well, whole cell just doesn't really tell you anything because you're controlling all of the inside of the cell, which I mean it's a bit dramatic, but I think I was really just trying to justify all the time and effort I put into perforated patch. But yeah, I mean, really what you want is the best electrical access to the cell while minimally interfering with the physiology of the cell. And I think a really good method for this is also using what's called Sharp's electrodes. And in this technique, you have a tiny micropipette. So normally the pipette has a resistance of around, let's say 3 to 10 mega ohms for neurons, because that's what I know, really depending on the cell type and everything. Well, with Sharp's electrodes, the resistance of the pipette is tens or hundreds of mega ohms. And you are therefore allowed to directly record electrical activity, but you can minimize how much you affect intracellular concentrations of the cell. But again, this technique is really difficult to perform. I've not really seen it done at all. And I think, again, it's like in the idealistic world where we can just talk to neurons directly.
Yeah, that would be great.
Oh, and then also another super cool type of recording is capacitance recordings. And this can give you direct information about the fusion of neurotransmitter containing vesicles to the membrane and their release. And firstly, it's been done at the calyx of Held, which is an extremely large synapse. And so you can directly patch onto that. And so when vesicles fuse to the membrane, they become a part of it. So there's more membrane. And this can be measured as an increase in capacitance. Capacitance is the amount of charge that can be held by a membrane. So if you have more membrane, more charge can be held by it. So it's an increased capacitance. And then when you have endocytosis, so when the vesicles part away from the membrane as they take neurotransmitter in, then the opposite happens and you have a decrease in capacitance. And so by performing this patch clamp and recording the capacitance, you can actually measure the endo and exocytosis of vesicles and neurotransmitter release directly, which I think is absolutely amazing.
Yeah, incredible.
And another thing you can do is relatively recent. It's called patch-seq. And this involves combining patch clamping with RNA sequencing. So you carry out first whole cell recordings, and then you apply a gentle suction after you've done all your recordings, and you can collect RNA from the cell and then isolate it and sequence it. And that way you can characterize what cell type you've actually recorded from, which sometimes isn't clear when you're targeting the cells. And you can combine the molecular and physiological information and data that you get from single cells and associate it with each other. So it's a really powerful technique to characterize the properties of a single cell.
At what point in the process do you collect the RNA?
So you carry out your recordings, and then as you're retracting the pipette, you take some of the cell with you. Mm-hmm.
Okay, so instead of imaging it, they do RNA sequencing.
Yeah, that's a good point, because another way that you can characterize a cell is by its morphology, and usually if you want to do that, you'd fill the pipette solution with a dye. And then when you're recording from the cell, because it becomes continuous, the dye fills the cell, and then afterwards you can carry out imaging to look at the neuron that you're recorded from, which is also amazing when you get to see the single neurons that you connected with.
Yes.
Can you tell that I like patch clamp? I've gotten so passionate.
I love it. I love it.
Okay. I also wanted to talk about the information you get when you're actually recording from the cell. Specifically, a really interesting way to really tap into the capabilities of a neuron is dynamic clamp. So normally when you are doing voltage clamp, the system is able to read the membrane voltage and then inject current to change the voltage to meet your requirements. In dynamic clamp, the system is able to instantaneously read the membrane potential, and then it generates a virtual current to be delivered. And this virtual current can be calculated based on an algorithm that represents specified membrane mechanisms that you want to test. So dynamic clamp is a really good way to test any computational hypotheses that you have in living cells. So for example, you can replicate the actions of a modeled voltage-gated ion channel, and you can use the system to read the membrane voltage, and then you calculate the value of conductance of your virtual channel that you want to implement in the neuron. And then the system multiplies that conductance by the difference between the membrane potential and the reversal potential of the modeled conductance. So ultimately, you are simulating the effect of having an ion channel in the neuron that may not actually exist in the membrane. I think that's a really cool way to test computational hypotheses experimentally. And it's also a good place to mention that a lot of patch clamp data can be really useful for computational models, because a lot of computational neuronal models are based on the biophysics of neurons. And so you can use the data to add it to create models, and you can use models to test them experimentally.
That's amazing. So as you were explaining that, immediately I started thinking like, oh, like perhaps they use some sort of like machine learning algorithm to be able to model all of the different parameters. And then you mentioned that like modeling plays a major part in it. And it's also really cool because perhaps it reduces the need of using drugs and stuff. Like you can kind of immediately test, like you mentioned, different parameters and like insert an ion channel or potentially block certain ion channels and that kind of thing. So yeah, that's super cool. Like within the same cell, you can run so many hypothetical experiments.
Yeah. And then I wanted to mention that a lot of what we've been talking about applies to recordings that are done from brain slices. So they're called acute ex vivo slices. So you can do this from a variety of animal models based on your research question, of course. But I think most commonly people do whole cell patch clamp in brain slices from mice or rodents. But we can also do patch clamp in vivo, so in a living organism. I've not had the opportunity to do in vivo patch clamp yet, but I think it's really amazing that you can do that without disrupting the brain significantly, because your pipette is tiny. So the techniques are the same as I've described, but of course, you're not really getting good optical access to the neurons that you're targeting, because in a slice, you can see all the neurons, you can look at the brain region that you want to get to, and you can find that region, look at a neuron. And also should mention that when you're forming that seal onto the neuron, a lot of it depends on the optics that you have. If you have the ability to look at the pipette in relation to the neuron, where it is, and whether it's pressing up against it, then that really helps with your seal formation. In vivo, you can't do that. So you have to do what's called a blind patch, which is targeting cells purely based on looking at the activity that you're recording from the pipette. So when you're approaching a cell, you're injecting a test voltage pulse and measuring current. And then, because I mentioned Ohm's law, you can then look at the resistance of the surrounding environment of the pipette, and that's how you can tell whether or not you're on a neuron. Yeah, so you have that blind patch clamping, but more recently, people are doing two other methods of in vivo patching. One is shadow patching, which is basically having really poor optics. You can kind of tell where you are based on the shadow of the pipette. I'm not going to go into the details of it, but it's like a rough estimate of where you are and whether or not you're approaching a cell. And also you can target cells by 2-photon imaging. So if you have cells expressing any fluorescent markers, you can target it that way. But it's nowhere as near as accurate as in brain slices. But yeah, of course, in vivo patching is really, really useful for looking at the cell activity during behavior or awake states even. And obviously, in a living organism, it would be a lot different too in a slice that is technically alive, but not working in the same way it would in an animal.
Yeah. And often like long range projections and all of that is all cut. So all of that information is lost and you
Yeah.
get that benefit with a live animal, but way more challenging.
Yeah. So now that we've gone over all the subsets of techniques under patch clamping, and we've also talked about how each one can give you different information about neurons, and we've also talked about how intuitive patch clamp is and all of the variables that need to be controlled, like the pipette size, the pipette solution, the optics, and the noise in the room or the temperature in the room or just everything. But let's talk about robots now.
Yeah, definitely. So Simon Schultz is a group leader at Imperial College London, and he's done a lot of work on robots and patch clamping. And I actually came across a poster from a PhD student in his lab at Fens, and that's when I first came across patch clamping robots because, as we've mentioned before, it's such an intuitive thing. I always thought that that's the last, if not at all, taken over by robots skill. Yeah. And anyway, they did it and they were
Yeah.
successful.
I actually saw that the first autonomous patch clamp robot paper was by a group at MIT, and they used it for in vivo recordings. But then Simon Schultz's group also contributed to this and made their own patch clamp robot. And most recently, there was a preprint in 2022, 2-photon targeted quad whole cell patch clamping robot by Gonzalez, Quare, and Schultz at Imperial.
Yeah, exactly. So, as we mentioned, in vivo patching is usually blind. And so, what they've done is that they've introduced a 2-photon microscope to target fluorescently labeled neurons. And then they combined this with the autonomic patching by having a closed loop system. And the way that they do it is they have a visual algorithm that allows the system to be able to target the cell in focus and iteratively adjust the visual optics while the pipette is approaching the trajectory. Because one thing we haven't mentioned is when we are going in with the pipette into the cell tissue, approaching the cell, oftentimes the optics do change, perhaps something to do with the focus or the consistency of the liquid in the tissue. And with the microscope, we can fine tune and adjust the focus. And their goal is to make all of it automatic. And so they initially really struggled with this visual component of patching, but they've solved it. They've opted for a closed loop system as opposed to an open loop control of pipettes and visual information. And I believe they use some machine learning visual algorithm to be able to keep on top of the differences in visual information.
And to do the robotic patch clamp experiments, Simon Schultz and his team did indeed use the Patch Star micromanipulator, which was provided by Scientifica, the sponsor of today's episode, which has a stable design and ultra-quiet electronics, which enable it to perform long-term patching experiments.
And then the second challenge that they face, which is the one that we've mentioned, is about the pressure. So they've developed this custom pressure regulation system, which allows them to fine tune the amount of pressure they want to apply onto the cells. And with this, they've been able to have, I would say, quite a high success rate for a robot. So they've obtained cell attach recordings on 47% of attempts at patching and 22% where they succeeded in doing whole cell recording. So 20% is, they still have quite a while to go, I think, to be able to really justify the costs, and which I imagine would be quite high when this becomes commercially available. But I think it's very impressive given that obviously robots don't have the kind of intuition that we mentioned. It's all based on pressure points and I guess feedback loops and just trying to find a cell. In terms of, I think, maintaining brain tissue intact, it may not be the best, the most optimal, especially if they're using live animals, just because I think in the MIT paper, the original one, they mentioned they had like a 9% yield of like over 100 attempts or something. So you can't go poking in the same brain 100 times to try and get a few cells, that would definitely damage the brain no matter how thin and small your pipettes are.
Yeah, that's exactly what I was going to say. Yeah, it was an improvement from the original 9% success rate. But I think the failure of robots is that you're not necessarily increasing the efficiency. So this is also something we should mention for anyone who doesn't patch, that the success rate is quite low. So depending on the cell type you are targeting, like for example, hippocampal or cortical, normal meaning like pyramidal or principal neurons, you would get maybe 0 to 10 cells a day maximum. Like in culture, maybe you could get more because you can also patch in culture neurons. But yeah, I mean, the robot is, I don't know how many, but maybe it's getting like a 20% success rate, but you need to attempt 100 cells. And so like the main positive attribute that a robot has is it can make more attempts because it just goes. It doesn't need to go drink water and it doesn't need to get food. But then, you might be damaging the tissue.
Yeah, absolutely.
Yeah.
I think it's super exciting. Like if they manage to increase the yield and if they perhaps become more efficient, like you mentioned, in terms of like it's not necessarily about patching 100 cells and getting 10, like more like patching 20, getting 10. That I would consider that more efficient. But I think the insight would be incredible, like the amount of data that we would get, especially in vivo patching, which is incredibly challenging as it is. I think we would have such an amazing insight into potentially neurons and dynamics of neurons that we don't know anything about due to the limitations of the challenges that come with this technique.
Yeah, and another amazing thing that robots could do is record simultaneously from multiple neurons. Like it could literally patch loads at a time, right? And then maybe you'd get at least 2 simultaneously recorded, and that can give us so much information about how 2 neurons or more communicate with each other. And in a normal rig setup, you could do usually up to 4 at a time. So yeah, it is definitely exciting, but not fully convinced.
Yeah, we still have our jobs for a little while longer.
So I think we'll wrap up today's episode there. I hope that you've enjoyed learning about patch clamp techniques. Thank you for supporting our podcast. Feel free to share it with your friends and follow us on Twitter and Instagram at neuroverse_pod. And also check out our website, www.neuroversepod.com, for more information on all of our episodes, all of our show notes, and all the links to everything we've mentioned in today's episode.
Yes, and today's episode was sponsored by Scientifica, one of the leading distributors of electrophysiology and imaging equipment. Do check out their resources for state-of-the-art equipment and more.